Thank you very much for having me. Very excited to be here. I'm Jonathan Thon. I'm the CEO, founder of Strm Bio. Strm is developing extracellular vesicles to deliver gene therapies in vivo, so non-viral gene delivery. And I think we all appreciate the value or the promise of gene therapy here, this idea that we can cure diseases as opposed to treating symptoms. But we also appreciate that there are some significant challenges as well. The delivery platforms we have to deliver those gene therapies, ideally in vivo, are cargo limited. By that I mean we're limited in the modalities that we can package into them, how much cargo we can get in. We're limited in the fact that a lot of these editors that we're interested in are getting larger and they don't fit into conventional delivery vehicles. And as we're wanting to think about more complex diseases, we're going to start needing to multiplex gene therapy assets into delivery platforms, and the platforms we have just aren't good enough. The platforms we have are also not specific. When we inject them into the blood, they go to the liver. We've got now third, fourth generation gene delivery platforms, targeted moieties. When we inject them into the blood, 90–95% of them go to the liver. We need to be able to break it away from that and target some of these other organs as well. And they're mostly single-dose treatments. I mean, we talk a lot about one-and-done therapies, but to be frank, one-and-done is really tail-wagging dog. We're coming in with single-dose treatments because we can't come in with a second dose or a third dose because of immunogenicity issues, toxicity issues. But we really need to be able to do so because we're really limited in targeting a very small fraction of diseases we want to cure. And the strategies we're taking to cure those diseases are also a very small sliver of what we could conceivably be doing. So what Strm is doing is developing a non-viral gene delivery platform that lets us do that. It's an extracellular vesicle-based platform. We can tunably load. We can put into these vesicles RNA, DNA, protein. We can multiplex assets into them. We can control how much cargo goes into each individual vesicle. We can get a little bit, 10 copies, a middle amount, hundreds of copies, a lot, upwards of 1,000 of copies, of very large cargo. We've gone up 9.8 kb plasmid DNA. We stopped there because nothing exists larger at the moment, but if it did, we could package it in there. These vesicles are HSC specific. When we inject them into the bloodstream, they go to the bone marrow, specifically long-term hematopoietic stem cells in the bone marrow. They bypass the liver entirely, and I'm going to show you that data. It's a pretty big deal. And we can repeat dose with this platform. We've shown this in mice. We've shown this in larger animal models. I'll show you that data as well. We can come in with a single dose, once a week, once a month dosing, and I think that's pretty important. It really opens up the landscape of what we could go after. It also lets us start playing with dosing in a way that's safer for the patients that are ultimately going to receive our therapies. Now, the source origin of the vesicles we're producing are megakaryocytes. Megakaryocytes are cells in your bone marrow that make platelets. They also make extracellular vesicles. Actually, more than 80% of the vesicles in your blood are megakaryocyte derived. In the body, these vesicles play the role of sentinel. They're picking up on environmental changes. They're circling back to bone marrow stem cells. They're reprogramming those stem cells. What we're doing as a company is we're really leveraging that innate biology to deliver gene therapies in vivo. Now, because we're focused on microvesicles, which bud off of the parent cell surface, they have unique properties that we could just capitalize on, and we do. For starters, they're larger. Ours average about 200 nanometers in diameter. They're very well conserved. It means they have very large carrying capacities. We can package a lot of stuff into them. They are innately immune privileged, which means we can repeat dose with them, and we do. We're really leaning into that for one of our first therapeutic indications of interest. And because they share membrane composition and protein composition with the parent cell, they have on their surface a number of different receptors that drive very selective targeting to long-term hematopoietic stem cells in the bone marrow. And you can see that here, bone marrow expression, allowing us to bypass the liver entirely. Now, this is an off-the-shelf batch process. It doesn't need to be matched to recipients. What we do is we'll source primary human hematopoietic stem cells from commercial suppliers like the Fred Hutch or the New York Blood Center, GMP-grade cells. We could then expand them, differentiate them into the parent cell, and from the parent cell, harvest the microvesicles and isolate them. Now, this becomes a bankable product that, when interested, we can thaw and load exogenously with cargo of interest. That cargo could be messenger RNA, it could be DNA, it could be protein. As I said, we have multiplexed assets into them. We put in a CRISPR-Cas9 with a single-stranded oligo and a guide RNA, works perfectly. And the idea here is that once you've loaded your drug product, you can store the product frozen. We've done stability studies out to 9 months plus at this point. Ship to site of interest, thaw at site of interest, inject directly into the blood, let the natural targeting drive biodistribution, fusion with stem cell in the bone marrow, delivery of payload, and let that payload impart therapeutic effect. In terms of scale, we've recently scaled up to 22 liters in batch. We're running these reproducibly right now, see a lot of between batch and within batch consistency. Each batch produces on the approximately 1 to the 12 extracellular vesicles per batch. That's approximately what we're projecting a clinical dose is going to look like for us for some of our lead indications. We can run, of course, these processes in parallel. We can move the process into larger volume receptacles when we're going to need to scale up. I mentioned the product is we can tunably load. We load exogenously, and because we load exogenously, this is very different than a lipid nanoparticle. We're not formulating a particle around a cargo. The particle exists. We're loading that cargo into the vesicle. What that means is that the process of loading does not change the biomarker expression of the vesicle. It doesn't change the size or the morphology. When we inject these vesicles into the blood, it doesn't change their biodistribution profile. It doesn't change their function. We could load RNA, DNA, protein. We could multiplex. I think very exciting is the fact that we can fine-tune how much cargo we get into the vesicle, because there are going to be indications where you want to put a little bit of cargo in and stoichiometrically match combinations of cargo. There are going to be other indications where you want as much as humanly possible, and it's important to be able to do that. It's a different dosing lever than how many loaded vesicles you inject into the animal, which we can do as well. Now, when we inject these vesicles into an animal, they will biodistribute to bone marrow stem cells. When that cargo is a messenger RNA coding for a luciferase protein, we inject into the mouse, we'll see biodistribution and protein, luciferase protein expression in the bone marrow. We don't see any of it in the liver. And that's really interesting. And I put here a picture of a lipid nanoparticle with a messenger RNA coding for the same luciferase, and you can see the difference. It's stark. This experiment was done with 10-fold less messenger RNA per vesicle, done with 100-fold fewer vesicles injected into the mouse. And when that cargo is a plasmid DNA, in this case coding for red fluorescent protein or an M-cherry, we can come in with a plate reader and track the vesicles through the bloodstream. We see them pass through vascularized organs like the liver. We see them accumulate in the bone marrow. We see protein expression almost exclusively in the bone marrow. When we collect those organs and we come in with confocal microscopy, we can see protein expression in the bone marrow. We see a little bit in the liver, in the spleen. It's a hematopoietic organ. We don't see anything in the liver. We don't see anything in the lung. When we translate those studies to a larger animal model, like a non-human primate, we see the biodistribution profile of the vesicles mirror what we're seeing in the mice. We see them accumulating in the bone marrow. We see a little bit in the spleen. When we load a plasmid DNA into these vesicles and inject them into the mice, and we come in with a qPCR to quantify where that plasmid DNA is, we find that plasmid DNA in the bone marrow. We find a little bit of it in the spleen. We don't see it anywhere else. And we can repeat dose with this platform as well. When we have gone in mice, the safety tox profiles have looked great. I'm not going to show you that because instead I'm going to show you the NHP study. When we go into a non-human primate and we inject once a week for extended period of time, we collect the blood. We don't see any changes in blood cell counts. We see no evidence of kidney or liver damage, see no tissue inflammation. The animals are clinically asymptomatic. They actually gained weight over the course of the study. We had to dose up to keep the dose consistent. And so clearly there's a lot that we can do with a platform like this. And what you're seeing is a focus on blood diseases. We're very interested in blood disorders. My background is from a hematology background. I really care about these disorders deeply. And I'm not going to get a chance to talk about all of them, but I did want to talk about one that is particularly important to us, and that's Fanconi anemia. Now, Fanconi is a pediatric disease. It's affecting young children. It's a disease where there's a mutation in a protein responsible for DNA repair. And what happens is that as these patients age, as they reach 8, 10 years old, they start accumulating DNA damage. But they can't correct that damage, and so the cells end up dying. The patient undergoes bone marrow failure. That ends up killing them. Now, we know from clinical work that expression of wild-type Fanconi anemia protein will address the phenotype. We know it works. The way these patients are treated today are through bone marrow transplants. There are obviously complications transplanting bone marrow to a patient, a child, undergoing bone marrow failure. What we think we can do, and the strategy that we're proposing here, is to load a messenger RNA or a plasmid DNA coding for wild-type protein into a vesicle and inject it into the bloodstream. The targeting would drive by distribution, fusion, and delivery to bone marrow stem cells, expression of wild-type protein in a way that bypasses the need for bone marrow transplants entirely. What's attractive about this approach is that we should expect to see protein expression in patients within the first 24 or 48 hours of administration, stabilization of blood cell counts within the first 6 to 9 months of initiation of clinical study, which is fast. What we also expect this to be is a repeat dosing strategy where we're dosing every 2 to 4 weeks. But what's exciting about that is that this can be a treatment for Fanconi anemia. It can also be a way of priming patients to get their blood cell counts up before they undergo bone marrow transplant as well, and I think that's pretty important. We do have a really strong potency assay for Fanconi anemia. It's called mitomycin C. It's an agonist that causes DNA damage, and if you have functional Fanconi anemia protein, the cell should be able to correct that damage and keep the cells alive. And in fact, when we load a plasmid DNA coding for Fanconi anemia protein and co-incubate Fanconi anemia knockout cells with these vesicles, we'll see protein expression. When we come in with a Western blot, we'll confirm that that protein is indeed Fanconi anemia. And when we run a potency assay, we can see that the cells remain viable, so the Fanconi anemia protein that we're producing is in fact functional. Now again, there's a lot that we can do with a platform like this, more than our company can conceivably do in its lifetime. We are interested in a select number of diseases that we think we're best empowered to go after first. But there are a lot more diseases out there that need to be treated. And what we have here, fundamentally, is a gene delivery platform. So if you have gene therapies, successful gene therapy programs that you want to translate in vivo, reach out. Happy to have that conversation. We're partnering now. I provide my contact information at the very bottom, so feel free to find me at this conference or afterwards. Thank you.